can't see them out there in outer space, so you can probably hear them. Anyway, I want to welcome everybody to the PhD defense of Orville seed bulbos, Pili grass seed dormancy, smoke-stimulated germination, and harvest timing. Orville started his academic career in 2002 with a graduation from the University of the Philippines, Los Baños. Came to UH in 2005 and did a weed control and native plants for establishment on the roadways. He finished in 2009. So between 2009 and today, he's been working on his PhD dissertation, and it's part of a larger project to look at producing native Hawaiian plants on the roadways. We have a Department of Transportation project whereby we have funding to look at producing native Hawaiian seeds on the roadways so that the roadways become a productive area for native Hawaiian seeds, and then those seeds can be used in wider conservation projects. Orville's going to give his presentation today just to give you some idea of the kind of effort that went into this. You're going to see a lot on seed germinations, seed storage, and he handled just about 76,000 individual seeds for his first project. So even though it's a little bit on the low-tech side, it's all seed germination and so forth, it's high-density quality information. So just imagine handling 76,000 seeds over the last 4 years. You get the idea of the level of dedication and detail that's what you're going to view today. So without any further ado, I'll introduce Orville seed bulbos, and Orville, it's all yours.
Thanks, Dr. DeFrank. So today I'll be discussing the results of my dissertation research on Pili grass, seed dormancy, smoke-stimulated germination, and harvest timing. So in Hawaii, there has been recent interest in the use of native plants in a number of purposes, such as restoration, urban landscaping, and roadside revegetation. While a number of plants have been identified and evaluated for these purposes, studies are needed on seed production and germination biology in order to increase availability of planting materials, as well as to develop large-scale establishment protocols. So Pili grass is one native Hawaiian species that is being expanded for use in different purposes. And Pili grass is a drought-tolerant native perennial bunch grass. In Hawaii, it is found on all main islands from the coast up to 700 meters above sea level. Pili grass is considered as an important revegetation species in Hawaii because it is adaptable to low rainfall and low nutrient soils, and it has cultural and ecological value. In fact, the native Hawaiians have used Pili grass as their thatching material for their hales, or their traditional huts. So in recent years, it has been expanded from restoration to water-efficient landscaping, stream bank stabilization, and buffer strip plantings. So what critical factors limit the use or availability of Pili grass seed? 3 come into mind. One is seed harvest timing. Second would be seed dormancy. And third would be seed storage. So these 3 themes, so let's go to the harvest timing first. Harvest timing is important for Pili grass because there hasn't been any protocols developed for Pili grass yet. Timely harvest is essential because if you harvest too early, you have more immature seed, while if you harvest too late, you lose seeds from shattering. Seed dormancy is another critical limitation since you can't use freshly harvested seeds, they're dormant. And this could be a problem in a revegetation sense because it can delay canopy fill-in and making your plantings more prone to erosion and weed invasion. Seed storage is another critical aspect. Knowing the right storage conditions is essential for preserving seed viability, and it also builds a critical mass of seeds for large-scale projects, and it also stabilizes the supply of seeds by buffering it. So these are the 3 themes of the dissertation. So this is a photograph of if I was to think about a shot of my dissertation in one photograph, this is how I would visualize it. So let's go to harvest timing first. What harvest timing indicators can be used for Pili grass? A number of crop and non-crop species have used growing degree days or growing degree units as a means to predict harvest timing. growing degree units considers temperature and heat units accumulated over time and relates it to seed development and maturity. GDUs can be calculated by measuring the maximum and minimum temperature for a specific day. And you take the average temperature and subtract it with a base temp. A base temperature is at the temperature at which growth stops. For Pili grass, it's about 15 degrees Celsius. So if the maximum and minimum temperature at one point in time or during one day is 15 degrees Celsius, you have a GDU of 0. So to make growing degree units a meaningful measurement for harvest timing, we relate it with spike and seed development. So for the harvest timing study, I collected spike and seed data and related it to average cumulative growing degree units across seasons. So this is how I did the experiment. So I harvested 40 stems per row in my Pili grass field, separated 30 stems or combs, collected seed yield for that 30. The remaining 10 combs were used for calculating the total spike per comb. And the total spike per comb were separated into number of intact, number of shed, and number of unopened spikes. The number of intact and shed spikes were then bulked to collect a sample of 10 spikes and measure moisture content of the spikes. Once the samples were dried, I collected the seeds, measured their dry weight, collected their number of seeds, and then separated it into filled seeds and empty seeds. In addition to that, I also calculated the average dry weight of seeds, and this is your measure of physiological maturity. Physiological maturity is defined as the maximum weight that a seed can carry. So the data collection, I had 2 seasons during the year. I have a spring season and a fall season. So for the spring season, I cut the Pili grass stands during the spring equinox, waited for 57 to 59 days to start harvest, and harvesting or data collection continued on until the end of the summer solstice, where I cut the Pili grass again, Pili grass stand. The fall growing season was similar. So I cut the Pili grass stand on September 22nd or 23rd, waited for 57 to 59 days, started harvesting, collected data until the end of the winter solstice. During the period between the first cut and the second or the last cut, I recorded the maximum and minimum temperatures every day and calculated the cumulative growing degree units. So the data was I collected data from 2011 to 2012. I did analysis of variance and regression analysis on the data. And this is just a short video of how I did the experiment. So I have my rows of Pili grass here, and I collected about 30 combs, or 40 stems, separated it into 30 and 10 stems. Look at that, so I'm counting per row. And for the 10 spikes, I separated the spikes into number of intact. So I recorded number of intact, number of shed seeds, and number of unopened spikes. Actually, spikes, not seeds. So results of the harvest timing indicates that growing degree units can be used as an estimate for harvest timing. It was consistent in 3 of the 4 cropping cycles and was observed in a number of data that I collected, including seed yield for 30 combs, filled seed number of 10 spikes, total weight and number of seeds, and average weight of individual seeds. So the optimum CGDU was between 768 and 778, which is equivalent to 79 to 82 days after cutting. This range of CGDU coincided with spike moisture content range, which is 0.68 to 0.72 grams of water per dry weight. So this is one example here. So you have seed yield of 30 combs, and you have spike water on this side. So this is the peak seed yield for the 3 consecutive cropping seasons. So if you draw a line with the same like GDU, you have a similar range of spike moisture content across the season at this point. In addition to the quantitative data that I collected, visual seed head tangling also corresponded as with the seed maturity data. So seed if you don't collect the, I guess, your moisture content, if you don't have a means to record your moisture content, then you can just look at the field and see if it's ready. So the tangling is actually a good means of measuring maturity. So here is the Pili grass field, successive photos during my harvest period. So you can see the spike development over time. Here it's dark brown and it's turning light brown as it begins to tangle. And in this set of photos, the maximum or the most intense tangling was observed at 755 cumulative growing degree units, or 0.75 grams of water per dry weight, which is close to the predicted value. In addition to finding an optimum amount of seeds or a harvest index, we also observed a decline in the Pili grass productivity over time. This was observed in data like seed yield from 30 cums and total spike per cum. So let's look at seed yield per cum, per 30 cums. So this is grams of seed, cumulative growing degree days. You have your spring 2011 season here, fall 2011, spring 2012, and fall 2012. So as you increase your cropping cycles, you can see a decline in the productivity of your Pili grass stand. So this is total spike number per cum. So y-axis is the total number of spike per cum. This is your cumulative growing degree units. You'll also see a decline over successive cropping seasons. You can see that fall is flat. So a number of factors can be caused by the decrease in productivity can be caused by a number of factors, including stand age, cutting height, thatch accumulation, and increased pest incidents. Further studies are recommended to tease out the effects of these factors. So now let's focus on the seed dormancy aspect. So as you can recall, freshly harvested seeds of Pili grass is dormant and it will not germinate. Pili grass seeds, according to Baskin and Baskin, possess a physiological dormancy. Physiological dormancy is the most abundant form of dormancy in seed-bearing plants, and it's classified into 3 levels. You have deep, intermediate, and non-deep. Pili grass likely possesses non-deep dormancy since it is responsive to GA or gibberellic acid application and warm and dry storage. So for this dissertation, I wanted to look more in depth and wanted to understand more about the mechanisms behind the dormancy of Pili grass seeds. So I looked at the components of non-deep physiological dormancy in Pili grass. So this is a Pili grass seed, a cross-section of it. You have an embryo-imposed dormancy, which is physiological, and a coat-imposed dormancy, which is physical. Coat-imposed dormancy is caused by physical restriction by the embryo, by the endosperm. So the endosperm is basically restricting embryo growth and radical protrusion. Coat-imposed dormancy can be overcome by increasing embryo growth potential or promoting endosperm weakening. Gibberellic acid plays a role in relief of coat-imposed dormancy because studies have shown that applications of gibberellic acid increases embryo growth potential and promotes endosperm weakening. So for this part of the dissertation, I wanted to examine the role of coats. So what I did was I scarified the seeds to mimic endosperm weakening. So here are my treatments. So the scarification study was done in March and in June harvested seeds. So I have 5 treatments. So my intact seeds would serve as my control treatment. I had naked caryopsis, top cut seeds, and spear cut seeds. These did not expose the embryo, but it increased water uptake. And I have the spear side nick seeds where I expose the embryo, which mimics artificial endosperm weakening. So for this one, I cut it on the on side. This one, I cut the spear side off. And this one, I sliced the seed diagonally to expose the embryo. So results of the scarification study. As you can see here, the spear side nick seeds, which exposed the embryo, exhibited consistent germination across the seed batches. The naked caryopsis and top cut seeds exhibited germination in the March 2012 seeds, but did not exhibit germination, so it was not consistent for these treatments. Spear cut seeds did nothing to the Pili grass. So percent germination of spear side seeds were consistent, and percent germination was 30% and 19.5%. This confirms the presence of coat-imposed dormancy in Pili grass and may suggest the role of GA in endosperm weakening. This result also suggests the presence of physiological dormancy because I didn't, germination was not as high as my tetrazoline viability test, because the tetrazoline viability test is a test wherein you measure, it's a chemical test wherein you measure how much germinable seed how much a seed batch is germinable. So if you scarify your seeds, you only have 30 to 19.5%. So there must be something else that's controlling the dormancy, and that is actually the physiological component. So in addition to confirming the presence of the coat and finding that there's physiological dormancy involved, I also observed seasonality in the depth of germination, wherein the March harvested seeds were less dormant than the June harvested seeds. So I did chi-square test of the treatments. So I compared March versus June. So I had 33 seeds germinated versus June, and this was significant. So in general, you can see that there's much more germination happening in the March than in the June harvested seeds. Now let's go to embryo-imposed dormancy. So I was interested in looking at the factors affecting dormancy relief in Pili grass. And this can be divided into two. One would be chemical factors, which is within the seed or within the environment. The other one would be physical factors brought about by environmental factors. Examples of chemical factors that affect dormancy relief are the ones within the seed, which is gibberellic acid and abscisic acid. And you have smoke cues, which is from the environment. Examples of physical factors are temperature and relative humidity. So let's look at the physical factors first. So temperature, relative humidity. So in Pili grass, a number of studies have shown that Pili grass requires a dry storage requirement or dry after ripening period. And they recommend, these studies recommend storage under dry conditions for 6 to 12 months. However, these studies didn't look at specific temperature and relative humidity and how that affects dormancy loss. So for my study, I looked at season, the effect of season, the effect of relative humidity, temperature, and storage duration on dormancy loss of Pili grass. So this is how I did the experiment. First, I collected clean seed, placed them in unsealed packets. So each experiment contained about 252 packets. These packets were then placed inside desiccators containing saturated salt solutions. These saturated salt solutions under room temperature produces different relative humidities. So lithium chloride produced 12% relative humidity. Calcium nitrate produced 50% relative humidity. And sodium chloride produced 75% relative humidity. So the seeds were stored under these relative humidity conditions for 28 days. And these are the equivalent seed moisture content. So they adjusted, the relative humidity adjusted the seed moisture content to 6% 11%, and 14%. So after equilibration, I sealed the packets to maintain seed moisture content. So now I have the three different, the seeds with the different relative moisture contents. So I stored them in three different temperature regimes. 10 would be under refrigerated conditions, 20 is your ambient laboratory condition, and 30 is another incubated condition. So we stored these different moisture contents at different temperatures over different periods in time, so from 0 to 12 months. After each incubation duration, I collected seed germination tests to determine dormancy loss over time, tetrazoleum viability tests to determine seed deterioration over time, and seed moisture content just to make sure if the packets did their job. So this experiment was conducted twice using March and October 2011 harvested seeds. So here are the results. Let's focus on seed germination first. So we have the equilibrium relative humidity on this column. So you have 12, 50, 75, 12, 50, and 75. Temperature, increasing temperature as you go down the column. And you have months in storage, 0 to 12. So let's look at the seeds that were stored at 10 degrees Celsius first. So you'll notice here the percent germination of seeds were low, less than 3%. So this indicates that storage at 10 degrees Celsius, regardless of relative humidity, maintained dormancy of Pili grass over the 12-month storage period. So now let's look at the storage at 20 degrees Celsius and 30 degrees Celsius. So the first thing that pops here is the germination in the month 3 of March harvested seeds. You have germination going on here, whereas in here there's very little germination going on. So this suggests that this March harvested seed batch is less dormant than the October harvested seeds. And then lastly, let's look at the storage from 6 months to 12 months. So you'll notice that cells are shaded green. So the greener the shading, the more germination you have. So based on this data over here, we can see that the optimum dormancy loss conditions were storage at 12% relative humidity at 30 degrees Celsius or at 50% relative humidity at 30 degrees Celsius. So let's look at the tetrazolium test results. Similar structure of the table, except I don't have month 0 for this one. So the main thing to look at here is the red shading. So the red shading, the deeper the red shading is, the more seed deterioration you have. So looking at these two tables, the treatment combinations which exhibited consistent deterioration over time were the ones that were stored at the highest relative humidity at 75% relative humidity and at 20 and 30 degrees Celsius. So to summarize, storage at 10 degrees Celsius maintained dormancy and viability for up to a year. There was an observed seasonality in March seeds, in dormancy, wherein you have your March seeds having less dormancy than October harvested seeds. Oops. The optimum storage conditions for dormancy loss was 12% relative humidity, which is equal to 6% seed moisture at 30 degrees Celsius at 12 months, or 50% relative humidity, 11% seed moisture at 30 degrees for 9 months. The tetrazolium viability test indicated that seed deterioration was observed in the treatments that were stored in the high relative humidity treatments and at 20 and 30 degrees Celsius. Let's go back to the factors affecting dormancy relief. So we have now answered the physical factors. Now let's focus on the chemical factors. So chemical factors involves plant hormones within the seed and smoke cues. So I did studies on plant hormones, and what I found out that if you increase the GA levels of the seeds above ABA levels, you'll have germination. So now let's focus on smoke cues, which is effects of carotenoid and cyanide. My smoke picture. So fires provide a recruitment opportunity for plants. They change the growing environment by altering the soil, increasing light availability, reducing allelochemicals, and removing plant competition. So fires provide a suitable place for plant recolonization because it creates a bare ground area. So in addition to physical changes in the environment, fires also produce chemical compounds which plants utilize for cues as germination. So burning vegetation produces tar and char, particulates, and condensates, which are deposited into the soil. These materials are then release a number of known germination stimulants called carotenoids, cyanide, nitrate, and nitrite. So these compounds stimulates germination in a number of species. Pili grass is reported as a fire-adapted species. However, studies on smoke-stimulated germination have been limited. The only study that I found was Campbell's dissertation with Pili grass, and he reported germination stimulation with aerosol smoke. So aerosol smoke is basically he's just burning the plant material and piping the smoke through the seed. So in this dissertation, I wanted to understand smoke stimulation more, and I also wanted to develop a smoke-based seed treatment, which can be an alternative to gibberellic acid. So studies on smoke-stimulated germination, I tested different smoke water solutions and the smoke-derived compounds. So for the smoke water solutions, I had Pili grass smoke water, xylo smoke water, and food-grade liquid smoke. Food-grade liquid smoke was included in the study because it is commercially available, so you can just buy out of the store and use it for your germination. Xylose was included because it didn't contain any nitrogenous compounds. For the smoke-derived compounds, I tested carotenoid and cyanide. So carotenoid was recently identified. So it was identified by independent groups, one from Australia, Dr. Flimati's work, and one from South Africa in 2004. And cyanide was just published a couple years back. So cyanide was also identified as one of the components in smoke that stimulates germination in plants. So what I did for my study was I did dose rate studies first on these compounds, try to determine the maximum amount of material for maximum germination. And once I found the dose rate studies, once I found the optimum concentrations, then I did some germination studies comparing smoke and the two active compounds. So for the smoke water studies, or for the dose rate studies, I did germination tests on March 2011 seeds. And for the smoke versus active compound study, I used July 2012 seeds. So here's a short video of how I did the smoke solution. So I preheated my flask with an open flame, and then piped 30 milliliters of air per minute into that flask. And once the flask has been preheated, I added 2.5 grams of material, either Pili grass or xylose. So you can see that it burst into flames. And now it starts producing smoke, so I quickly closed it. And then the smoke was piped into 100 milliliters of distilled water in an Erlenmeyer flask. So results of the smoke-stimulated germination indicated that, that this is for the dose rate studies. For Pili grass smoke water, you don't need any dilution at all. Xylose also didn't need any dilution. For food-grade liquid smoke, you need to dilute it to 1% by volume. And for carotenoid, I tested concentrations between 0.0067 to 66.7 micromolars, but didn't see any response in carotenoid. See any response actually in Pili grass. So this may indicate that KAR1 is not the compound responsible for smoke-stimulated germination in Pili grass. And then I tested cyanide with potassium cyanide as my source and found stimulation, significant stimulation between 50 and 500 micromolars. So now that I've optimized, or I found the optimum dilutions, I did a study comparing the smoke water versus the 500 micromolars of cyanide with July 2012 seeds. So here is my data. So I have percent germination on the y-axis. And here, 1 and 2 corresponds to runs 1 and 2, because I repeated the experiment. And they were separate because there was a significant run interaction. So I have distilled water, 500 micromolars potassium cyanide, food-grade liquid smoke, Pili grass. Neat means it was undiluted. Neat xylose also. So what you'll notice here is that the Pili grass smoke water and food-grade liquid smoke was similar in terms of germination stimulation with 500 micromolars of potassium cyanide. Xylose smoke water did not stimulate the seeds in this seed batch, but in the previous batch, the March 2011 seeds, had significant stimulation. And this may indicate some sort of seasonality going on with the smoke response. I did a follow-up study curious about how much cyanide is the Pili grass water containing. So I tested it, and it turned out positive, about 100 micromolars of cyanide. So within that concentration, that's stimulatory to Pili grass. I tested xylose smoke water and food-grade liquid smoke water, and they didn't contain cyanide, because if people will probably complain or something. So aside from that, so the germination response that we found in the xylose and food-grade liquid smoke may suggest that there are other non-cyanogenic compounds that promote seed germination in Pili grass. So in conclusion, harvest timing in the harvest timing study, we've identified the range of cumulative growing degree day units for optimum harvesting. We can use spike moisture content for monitoring our optimum, when to harvest our seeds. And if you don't have a means to measure moisture content, you can just use seed head tangling. So this is how the seed heads would look like if it's tangled already. In addition to finding the optimum seed harvest timing, successive cropping reduces crop yield. There was an observed decline in the Pili grass stand over time. The results of the scarification study confirms the role of coats and suggests the role of gibberellic acid in endosperm weakening. And there's also an indication that physiological dormancy is going on. There was also an observed seasonality wherein the March harvested seeds were less dormant than the June harvested seeds. For the optimum dormancy loss condition study, seed viability was maintained for up to a year with storage at 10 degrees Celsius, regardless of relative humidity. There was a seasonality effect. We saw that March harvested seeds again were less dormant than October. The optimum storage conditions were storage at 12% relative humidity, 6% seed moisture at 30 degrees Celsius for 12 months, or 50% relative humidity, or 11% seed moisture at 30 degrees Celsius for 9 months. The tetrazolium test indicated that viability loss was observed in the high relative humidity treatments, which were stored between 20 and 30 degrees Celsius. So lastly, for the smoke-stimulated germination, cyanide was confirmed as one of the stimulants in smoke, and Pili grass smoke contained approximately 100 micromolars of cyanide. I tested KAR1, carotenoid, which was another compound that was recently isolated in smoke and didn't stimulate Pili grass, so that's not the compound responsible. And the germination that was observed in the smoke solutions without cyanide indicates that there are non-cyanogenic compounds that exert an effect on pili grass. So practical applications of this dissertation. So for seed production, you can use CGDU, spike moisture, and seed head tangling as your harvest indicators for timing your harvest. Should aim for harvesting in March, so as to minimize your depth of dormancy. So it's shorter in that high temperature storage treatment. So to lose dormancy, you should store your seeds at 6% moisture at 30 degrees Celsius for 9 to 12 months. And then after you lose your dormancy, you should store your seeds at cold temperatures between 5 to 10 degrees Celsius to maintain viability. For field establishment, we found that smoke water can enhance seed germination. We can apply smoke water by pre-soaking the seeds or spraying or applying it as a hydromulch additive. So aside from the practical applications that were found out in this dissertation, there are also ecological implications. So the results of the study can explain why pili grass is found in certain areas. And it also explains the ecophysiology of pili grass. So the hot, dry areas where pili grass is usually found is conducive for dormancy loss. So here's a map of the state of Hawaii. So the areas wherein pili grass is found is usually on these red areas where you have very dry and hot conditions. So the dry leeward sides where pili grass is found experiences a distinct wet and dry period. And these distinct periods provide an annual cycle of seed production and dormancy loss. So you have, during the wet season, you have plant growth and seed set of pili grass. After the wet season, you have the dry season, which is conducive for dormancy loss. And once the dry season is over, then you have the wet season coming in, and your seeds have lost dormancy, and you have germination. In addition to that, the smoke-stimulated germination studies indicate that pili grass exploits fire for persistence and dominance. So fires create a bare ground area wherein you have good growing conditions. You exclude competition, increase nutrient availability. And what pili grass does is it utilizes the smoke cues for increasing seedling establishment or recruitment. So in other studies have found the stimulatory effects of fires in pili grass stands. This study by Campbell indicated that you can rejuvenate pili grass with burning. And here in Hawaii, there has been historical accounts that the native Hawaiians burned pili grasslands to stimulate new growth, which they use for thatching. Well, I'd like to thank a number of people because this dissertation isn't possible without the help of a huge number of people. So I'd like to thank my advisor, Dr. Joe DeFrank, for providing the technology for everything, for streaming this dissertation. Actually, without him, the streaming thing is not possible. Dr. Criley for suggesting smoke when I was still starting out in my dissertation, trying to find some interesting studies to include. Dr. Daler for the scarification study. Dr. Kobayashi and Dr. Paul for providing equipment and chemicals. I'd like to thank Dr. Tessie Amore and Peter Toves also, since they were the ones who, when I didn't have enough funding, they were the ones who took care of me. And they were also helpful, they were very helpful in terms of providing lab space and equipment for my research. The people from Magoon Research Facility, Craig, Neil, and Ronald, for my field studies, they were very helpful for, like if I needed planting, they're there to help me plant my pili grass. Dr. Gavin Flemati of the University of Western Australia, he was very helpful since he gave me samples of KAR1 to test. Scott Lucas and Kai Umeda, who was a visiting faculty in our lab. Scott was very helpful in my field studies and very helpful also in setting up all of the equipment for this video recording session. Dr. Matthew Kramer and Dr. Dong Mei Lee for statistical advice. Dr. Dong Mei Lee was I asked for help earlier on, and then when I met Dr. Kramer was very helpful in my stats. The Sustainable Organic Farming Systems Lab and the Tropical Forestry and Agroforestry Lab for lending their weighing scales, because I didn't have weighing scales at that time when I was measuring my seed moisture content. The Chain Lab in the chemistry department, where I got samples of benzaldehyde and had the benzaldehyde cleaned for my germination tests. Chester and Wesley Yoshida. Chester is another PhD student and in chemistry, and I had several I guess meetings with him, like in terms of suggestions on how to do stuff in terms of chemistry. People from the NRCS Plant Material Center, because Glenn Sakamoto, David Duvachel, Kenny Reyes, Andres Huario, and Robert Joy, who's already retired. These guys gave me the planting material, so we collected the seeds using a combine for my study, because I needed a lot of seeds for this dissertation. Chris Dacus was also very influential, because he was the guy in HDOT who started all of these native plant research in Hawaii. Dr. Russell Yost for some statistical advice also, and Susan, Shirley, and Jeff for the clerical support. Kawahi and Martha for the stats, like giving me, like, because I was always I guess ranting, or I had statistical problems, so I had to like just to blow steam off, I guess. Dr. Bingham for preliminary consultations with the smoke study. Lori for the support. Colleagues, friends, and family, thank you, mahalo, and salamat. So this dissertation used about 76,600 seeds. So if you have any questions, thank you.
So have you rejuvenated the Pili grass plot at Magoon with smoke yet?
Not yet.
And how would you do it?
Well, I can do controlled burning, I guess.
Well,
But
I was wondering if you would burn it, or if you would spray on your smoke solution, or just what?
well, because one factor that may be implicate, or one probable cause might be the accumulation of thatch. So I need to remove the thatch layer. So it's not only the smoke application that can help. I need to remove some stuff there that might inhibit growth.
In the official Association of Seed Analysts, they often treat the seed with potassium nitrate solution to stimulate germination. Did you do any potassium nitrate examination of your seed?
At this point, no. I was just focused on the KAR and the cyanide.
I guess I'm curious, why did you select smoke over heat itself as your?
Well, I did studies. I did preliminary studies with heating, but I didn't get any response at all.
So in the field, if you wanted to use that smoke water, how would you do that?
So if you want to make, I guess you just follow the procedure and just have it on a large scale, like 2.5, and then scale it up. Or there is actually commercially available smoke solutions that you can buy a gallon, because There's one company in Australia. It's called Regen. So you can buy it commercially also if you don't want to make your own. Any more questions?
Okay, if there's no more questions, thank you, Orville, and we'll have the committee reconvene in about 5 minutes. Thank you very much.
All right. Thanks.